Hello students, welcome to the new lecture, and in this lecture we are going to discuss about the phenotype, genotype and also dihybrid cross. Okay, in the last lesson we have discussed about the monohybrid cross. So now we will discuss and we'll start with the phenotype. Okay, if you have any doubts while the lecture is going on, so use the comment section so that you can ask and we will clear about your doubts. Okay, so first phenotype. What is meant by phenotype? So phenotype, it means these are external visible characters. For example, if I give you a pea seed, okay, you can see just by looking the pea seed, you can say whether the seed is yellow colored or green colored or round shape and wrinkle shape. So it means just by looking, you can say what shape it is and what color it is. Okay, that is known as a phenotype. Okay, so this is known as external visible characters. External means on outside, so what the seeds have. So visible means you can see that these are the characteristics. What are the characteristics? Whether it may have the yellow color or it may have the green color. So these are the characteristics. Okay, so yellow, capital Y small y means what does it mean? It is nothing but yellow colored. Okay, this is nothing but yellow colored. Okay, so next in F2 generation, the 75% will be the yellow colored. So in the monohybrid we have seen, so in F2 generation by pollination, self pollination of the F1 generation, you will get the 75% yellow, 25% is green seeds, and also you can observe the external characters of those seeds. Okay, this is known as phenotypic ratio. So phenotypic ratio means how many are yellow colored and how many are green colored. So in that we got the value that is 3:1 ratio. Okay, what is 3:1? 3 is nothing but yellow colored. Okay, this is nothing but yellow colored and 1 is nothing but green colored. So what does it mean yellow color? So yellow means it is going to appear yellow in color, and green means it is going to appear the seed will appear in green in color. Just by looking you can say, but you cannot say what type of allele is present in it. Okay, you cannot say. Okay, now that is known as genotype. So genotype is nothing but these are the characters that are present in it. Okay, so in this genotype the characters are not visible in an organism. External characters you cannot say. For example, if I give you a yellow color seed, let us consider this is a yellow color seed. Just by looking at it, you can say this is yellow. Okay, you will say that yellow color. But can you say what type of genes present in it? It means what type of allele is present in it. What is meant by allele? Allele means a pair of genes. Okay, a pair of genes. So those genes are nothing but capital Y, or it may be small y, capital Y, or it may be small y. So can you say which type of allele is present in this yellow colored seed? You cannot say. Okay, why? Because the genes are in the cell of a seed, in the cells of a seed, so you cannot say. Hence we can say the phenotype of a seed, but you cannot say the genotype of the seed just by looking at it or just by looking the characters. So these characters are not visible externally. Okay, and in the genotype we have seen the F2 generation of the monohybrid cross. We have seen the 75% of the seeds is yellow and 25% are the yellow only. That is also homozygous. Homozygous means the same genes. You can see capital Y, these are the homozygous. Why it is homozygous? Means this gene and this gene, they are capital Y look similar. That is known as homozygous. So next will be the 25% will be the heterozygous. So how the 25% is heterozygous? You can see capital Y and also small y. So the hetero means different, homo means same. Okay, here we are calling it as a heterozygous. Why? Because one is a capital, one is a small, hence it is known as heterozygous. And 25% will be the small y capital Y or capital Y small y. This is also heterozygous. Okay, hence in the genotype of monohybrid cross, you will get the ratio 1:2:1. Okay, one is the homozygous and this is also homozygous yellow, and this is the homozygous green, and this is the 2 is the heterozygous seeds or heterozygous the seed or generation. So hence this is the genotypic ratio, or you can call it as a monohybrid genotypic ratio. Okay, so here we can see the example. Here we can see the example, the parents. So by extracting the gametes of the parental plants, you will get the uh, combination here, the capital Y, and also on the other side also the capital Y. So if you do the combinations, you will get capital Y, capital Y, capital Y, capital Y, capital Y, capital Y, and also capital Y. So 100% of the seeds are yellow only. 100% of the seeds are yellow. So this is nothing but, so by doing the capital Y genes. So if you take other gametes, that is a capital Y, small y, and also here is the capital Y, small y, you will get 3:1 ratio. What is meant by 3? 3:1 ratio is nothing but 3:1 means the 3 will be the yellow, okay? 3 will be the yellow and 1 will be the green, okay? You can see. So this is the phenotypic ratio. So what is genotypic ratio? The capital Y is how many? 1. So the small y, capital Y, small y is how many? The 2, okay? And small y is how many? That is 1, okay? Can you see? 1:2, 2:1, okay? This is the genotypic ratio. This is the genotypic ratio. You cannot say just by looking the seed, okay? It is present in the cells of the seed, okay? So but you can say how many seeds are yellow, how many seeds are green. That is known as phenotypic ratio, okay? And if you see that, if you take the similar type of the gametes, the similar type of gametes means small y, and here also the small y. In this case, all are same type of genes or similar type of the genes. So in this case, you are going to get, so the same recessive one only. Recessive means, so that is not dominant. So hence you will get all small y, and also the small y. So what does it mean? It doesn't mean that nothing but 100% is the green, okay? 100% is the green seeds, and also 100% the genotypic ratio is 100% small y only, okay? So this is the phenotypic and also genotypic ratio, okay? And this is about phenotypic ratio and genotypic ratio of the monohybrid cross. Now we will see dihybrid cross. First of all, what is meant by dihybrid? Di means nothing but 2, okay? Di means 2. Hybrid means the different type of genes, okay? That is for the different characteristics. That is known as dihybrid. Here we can see the any organism or any seeds that contain the 2 pairs of contrasting, okay? 2 pairs of contrasting characters, okay? So what are those characters? We are calling it as 2 pairs of contrasting characters. One character is for the color. What type of the color? It may have the yellow, okay, or it may have the green, okay? So next, structure or the shape will be the round, okay? The shape will be the round, and here the shape may be wrinkled, okay? So you can take, this is one characteristics and this is a second characteristic, okay? The first characteristic may be for the color. This is for the color, and this will be for the shape, okay? What shape it has, okay? We are calling this such kind of combination or the cross, such kind of cross between these 2 type of the genes, that is known as 2 pairs of contrasting characters. You can see here the color and also the shape are taken into consideration, okay? That is known as dihybrid cross. What is meant by dihybrid? Dihybrid is nothing but, so any seed or any parent plant that is having the 2 pairs of contrasting characters, and that is known as dihybrid cross. Example, the plant with a pure yellow and also round seeds, okay? Here we can say, here you can take the pure genes, okay, or pure plant or parental plant. So what is the pure parental plant? Is nothing but it is going to have all dominant genes only. Here will be that capital Y is the dominant. This is the yellow, and it is going to have the capital R. This is nothing but for the shape. So capital Y means what? Capital Y is nothing but, so yellow, okay? Small y means what? It is green, okay? So next, R means what? Capital R means round, okay? So next, small r means what? Small r means wrinkled, okay? So by taking this into consideration, can you say what is the shape of it? Its shape is nothing but yellow round, okay? This is nothing but yellow round. So take the another parent. So this is pure homozygous dominant. Okay, pure homozygous dominant, yellow round, yellow because capital Y is there and capital R is there, yellow round. So next take the another type of the parent, that is also, that should be pure, small y, small r. So what does it mean? It is also pure, homozygous recessive, green wrinkled. Okay, so these are the parents. So now take the gametes. If you take the gametes from the parents, what type of gametes you will get? You will get some kind of gametes that we will discuss, so that after a moment. So see here, on cross pollination of the pure, okay, cross pollination means between these two parents cross pollination should be done. So cross pollination of capital R, capital R, and small y, small r, you will get the F1 generation. Okay, F1 generation means what? F1 means filial one generation. Okay, so whatever the plants you will get by crossing these parents, that is known as F1 generation. So in the F1 generation, all are yellow and round seeds. We will see how the yellow and round seeds you will get. Okay, so self-polished F1 generation produces F2 generation with seeds of yellow and round seeds, that is nothing but having the combination of capital Y, capital R, or capital R. Okay, and yellow round like in this way and this way. So now do the cross pollination. Okay, cross pollination between the parents. So what type of gametes you will get? You will get these gametes. Those gametes are nothing but capital R, capital Y, capital R, and also capital Y, capital R. This is from one parent, and this another parent, like small y. Okay, small y, small r, and also small y, small r. Okay, so small y, small r, small r. Okay, here we can see the gametes. So here you can see the gametes. Okay, and this is from one parent, this is from the father, and this is from the mother parent. Okay, so you can see now what type of combinations you will get. Okay, you will get capital Y, capital R, small r, capital Y, capital R, small r, capital Y, capital R, small r, capital Y, capital R, small r. So it means 100%. The phenotypic ratio is 100%. The phenotypic ratio is 100% yellow round. How yellow round? Here you can say capital Y, capital R, small r. So what does it mean? Okay, this gene and also this gene, two type of genes are there. Okay, they are, one is for the color, one is for the shape. So it is yellow. Why? Because in this pair the yellow is there, the capital Y is there. If there is capital Y, the yellow color gene will express. Okay, why it is called? Why? Because it is dominant. The small y is green. Okay, it will not express. Why? Because the small is recessive, so the green color will not express, only yellow color will express. In the same way, the capital R, small r leads to the production of the round seeds only. Okay, round only, and the small r is for the wrinkle. Okay, so why the wrinkle will not express? Why? Because the dominant one is the round. Capital R is the dominant, it is for the round, and small r is the wrinkle, so it is not going to express. Hence we can get the seed yellow round only. It means all the progeny, this one is the F1 progeny. F1 progeny will have the capital Y, capital R, small r, every individual having the same type of combination. So it means all seeds in F1 generation are yellow and round only. Okay, so phenotypic ratio is 100% yellow round. Here you can say 100% yellow round, and the genotypic ratio is 100% capital Y, capital R, small r. Okay, so this is the phenotypic ratio. Now what you have to do? You have to take the parent, you have to take the individuals. I'm going to take this capital Y, capital R, small r, and this capital Y, capital R, small r. So now I will take these individuals as a parents, and I will do the self pollination in them. Okay, so now see here, we have taken capital Y, capital R, small r, capital Y, capital R, small r. So we have taken. Now you have to extract the gametes. How do you extract the gametes? See here, capital Y, capital R, small r into, it means this into is nothing but So some kind of pollination, whether it may be cross pollination or self pollination. So now capital Y, capital R, small r. So here how do you take the gametes? In this way, capital Y, capital Y, small r. So next capital Y, small r. Next small y, capital R. Next small y, small r. So how many gametes you will get from one individual? 1, 2, 3 and 4. 4 type of the gametes. Okay, you have to take the 4 type. We got the 4 type of gametes from this parent. If the parent, this parent and this parent both are similar, so you will get a similar type of genes. Okay, what are similar type of the gametes? What are similar type? Capital Y, capital R. Next capital Y, small r. Next small y, capital R. Next small y, small r. So you will get the same type of gametes. Okay, same type of gametes from this parent also. So this is the male parent, this is the female parent. Okay, so hence you will get such kind of combination. Here you can write male parent and female parent. So see the combination. So 1, 2, 3 and 4 type of gametes. 1, 2, 3, 4. Here also 1, 2, 3, 4. 4 type of gametes here we have taken in the checkerboard. Okay, or Punnett square. So these are the gametes that you will get. Okay, now do the combination. Okay, if you do the combination, you will get all these. So there is a small trick for you. Okay, there is a small trick for you to make the combination much easier. Okay, how do you take? So first write the diagonal way. Okay, in this way, diagonal. Okay, so if you take in this diagonal way, so all individuals in this from the left to bottom to the top right individual, all they have the same combination. See here, capital Y, capital R, small r. So I have highlighted the individuals with yellow color in the diagonal way. So this one and this one, second one, third one and fourth one. So these individuals are similar. So this is only the reason I have highlighted them with a red color. So individuals are same. So they have the same type of color, same type of genes in them, or same type of alleles present in them. Okay, so by this way you can save a lot of time while doing the combination. Not only for this, any type of combination you can follow in this way. So now come to the bottom one. So right after it you have to write this one. How do you write? Okay, so capital Y is there, so small y is there. So capital Y, small y. So here the small r and also small r. So small r. So this one and also this one. So this last and also this last. So both of them have the similar type of genes. Okay, here will be the come to next bottom. So this one and also this one. So both of them have the similar type of genes. Okay, small y, capital R, small r. Here also small y, capital R, small r. So by this way you can save a lot of time while writing. So you just check the what type of genes, what type of combinations is present in it. The same type will be present in the opposite direction. Okay, in the opposite. So now write this one. This is only one type of individual present in the entire table. Okay, that is small y, small r. Here we can see small y, small r. So you have written the small y, small r. Okay, here come here. So here the capital Y, capital R, small y, capital R. Okay, hence you will get capital Y, capital R, small r. So same type of combination. This same type of combination is present on the opposite side. So this corner means, so opposite will be here. Okay, opposite will be here. But in the middle there is another one. There is another one in the middle. So forget it about it now for now. So now come here next. Okay, this corner and also next corner here will be. So both of them have the similar type of combination, similar type of color and shape. So capital Y and also capital R, small r. Just by looking at it at these parents you will get it. Okay, so here also the same, both of them are same. So now come to that top right corner, okay, top right corner, that is capital Y is capital R small r. So this is only the unique one thing. So this will be the one and this will be the another one, okay. So now we have left over this, okay, this is only the middle one is left over. So capital Y small r and also capital Y, the small y capital R, okay, capital R. So this is the combination that you get, okay. So this is the procedure how you would do it. Okay, first let us take the same thing, okay, same thing, and we will write on our own, okay. So what are the combinations? Parental combinations, capital R small r, okay. So next will be the capital R small r, capital Y small r. Next will be the small y capital R, okay. Next one will be the small y small r, okay. So these are the combinations. Now the same type of genes also present on other side. So capital Y capital R, next with the capital Y small r, next one will be the small y capital R, next small y small r, okay. So whatever the combinations you will get. So first the diagonal, go for the diagonal. So capital Y small y, okay. So next capital R small r. So next come to here. Just now I said no, diagonal from the bottom left to the top right, okay. So next same type of combination, capital Y small y, okay, capital Y small y, next capital R small r, same type of combination here, capital Y small y, so capital R and also small r. So next capital Y small y, okay. So next here small r. So can you see here the diagonal, every gene here is the diagonal is same, so from the bottom left to the top right, okay. So now next come here, come here, capital Y, okay, so small y, next small r. So come to the opposite side, okay. Opposite means here you should come capital Y small y small r. See here combinations are same, okay. Next come here, next how you should write. So small y, here will be the small y. So next capital R small r. So you can see here, so combinations are same. So fill it up on the opposite side. Here will be, you should fill it small y capital R small r, okay. So this will be unique. So only one will be there here, only one type of individual. So that is all are small r, small y, small r, okay. Now come to the opposite here. You should come to this one, okay. So capital Y small y, next is the capital R. So go to the opposite here. Opposite means here, okay. So same capital Y small y, capital R, okay. Now come here. So how do you fill it here? Capital Y, capital R, small r. Up come to the opposite, capital Y, capital R. Okay, now come here, corner. Only one type of individual over here. Only one type. Okay, that is all are capital Y, capital R. So this is one. What is left over? This box is left over and this box is left over. So hence you can write the small y, capital R. So small y, capital R. So do the parents, see the gametes over here, okay. So next, so this gamete and also this gamete, okay. So next come here, capital Y. So small r. So compare this table, compare this table and also compare this table, both are same. So just I have shown you how to write the individuals simply, okay. So by with a small such kind of, with a simple trick, you can do the table within short period of time and you can know the combination, okay. So this is the male parent, this is the female parent. So the phenotypic ratio, check the F2 generation. This is known as the F2 generation. Now see the combination. What type of gametes and what is the size, what is the shape of the seed and color of the seed. So phenotype, that is nothing but if you count entire table, the seeds will be having the yellow round, green round, yellow wrinkled, and also green wrinkled. So count it. Count the number of individuals in this table. How many are yellow round? How many are yellow round? Only one. Okay. So this one only the yellow round. Okay, yellow round. And this will next will be yellow round. This is one, and this one will be the yellow round. Okay, and so on. If you count everyone in this individual, the yellow round will be 9. Total 9 individuals are there in this table. 9 yellow round. Okay, and it may have the any type of genes, whether it may be capital Y small y or capital R small r, anyway. Okay, but if you count every total individuals will be 9. Okay, why? Because the phenotypic ratio is 9:3:3:1. So 9 indicates that 9 is the yellow round, and next 3 is going to indicate that the 3 is a green round. Count, if see the table, in this table, entire table, the green round will be how much? 3. Okay, green round will be 3, and yellow wrinkle will be 3, yellow and wrinkle shape. So that is 3, and also green wrinkle will be 1. So just now I say this is only one type of individual with green. Okay, and this is the one type of individual with homozygous dominant genes yellow and red. This is the only one individual. Okay, so this is the phenotypic ratio. Now come to the genotypic ratio, means what are the types of alleles present in these individuals, that is known as genotypic ratio. Okay, genotype of F2 individuals, that is 1:2:1:2:4:2:1:2:1. So again we have a small trick to remember. Okay, just remember the 12 and 4. Okay, 12, 12, 4. Okay, now write these in reverse direction. After the 4, write it in the reverse direction. That is 12 means reverse direction means what? 21. Okay, 21 and also 21. So put the columns here. Put the columns between each and every number. Okay, if you put the columns between each and every number, what you will get? So this ratio will get 1:2:1:2:4:2:2:1:1. So compare here. Okay, compare this ratio and also this ratio. Have you got it? It is the same. Okay, it is the same type. So by using this trick, you can write in this way. So if you count, if you see the number of individuals, how many number of individuals with capital Y, capital Y, capital R, only one individual will be there, that is on the top. So for your easy understanding, we have highlighted with the color. Okay, capital Y, capital Y, capital R, capital R. So yellow. So next will be the capital Y, capital Y, capital R, capital small r, that is 2. So again go for the middle one. Okay, from the go for the diagonal way, from the bottom left to the top right. Okay, count this. How many are? 4 are there. Okay, first simply in the middle write capital Y, small y, capital R, small r. So that will be the yellow round, that is a 4. Okay, middle is over. So it means this individual is over. So next come to this. Okay, above the diagonal means this one. Okay, above the diagonal means this one. So write this combination above the right one, right colored highlighting. So above it means this one. Count how many. So this one will be there 1, and opposite side also 1. Okay, how many total? How many? 2 are there. So highlighted with similar type of color with 2 individuals, for the 2 individuals. So this will be the yellow round. How many are there? 2 after counting in the table. So now next to the come to the next one, that is blue one. So come to the middle. Okay, between this one and between this one, come to this. Okay, that is the capital Y, small r. So how many are there? Only 1. We have highlighted with blue one. Only 1. So we have written 1. Okay, next come to the this table. Okay, so this one, this column, this space. This is capital Y, small y. That means the capital Y, capital R, small r, and opposite side also capital Y, capital R, small r. How many are there? Only 1 on opposite to each other. Okay, so how many are? 2 are there. So write 2. Okay, 2 is over. So next come to the top right top left corner, that is capital Y, capital R, small r. So there is nothing to write on the opposite. So this is only the one type of individual with this combination. What is that combination? Capital Y, capital R, small r. Okay, how many are there? Only one. So write only one. So next, do the same thing. Come to the below the table. Okay, below the diagonal individuals, that is capital Y, capital R, small r. So how many are there? Two. This one, and opposite to this will be the another one. Okay, this will be the another. Okay, opposite to each other. Okay, this one and also this one. How many are there? So two are there. Okay, two are there. So right here 2. Okay, so next will be the green round. Okay, so green round will be how many? So here we can see, so this one green round will wait. So green round will be 1. Okay, so capital Y, small y, capital R, small r, that is only 1. Here we can see the one individual. So one individual will be here, small y, capital R, capital R. So only one individual. So next, come to the another, the green round, that is capital Y, small y, capital R, small r. So here will be the capital small y, capital R, small r. So this is one, and opposite to each other, this is one. Okay, opposite to this one. So how many are there? Are 2. Okay, so right here 2, right here 2. And next will be the, come to the bottom one. Okay, bottom one. So this is the bottom one. Okay, how many are there? Only 1. Okay, that is only 1 will be there here. Okay, only 1 will be there here, and also green round also only 1. So just by looking at this table, you can say, for your clear understanding, so we have highlighted with the colors, so and you can go for it. Okay, and this will be the structure. Wait a minute. Okay, so I hope this is helpful to you, and if you have any doubts or questions, and post it in the comment section so that we can clear your doubts, and in the next lecture we will discuss the next topic. Okay.